Hello again. Now we are at quiz number 3. Okay, question number 1. Which of the following statement is not true about mutations? If mutation occurs in a somatic cell, the mutation phenotype will be passed on. That's not true, because only the mutation in germ cells that are going to be passed on. Intrinsic mistake frequency of nucleotide selection during DNA polymerase. That one is about 10 to 1. That's correct. Point mutations involve changes in. That is also correct. Mutation refers to both. Well, the answer is A, right? Because it occurs in somatic cells, so it will not be passed on to the offspring. Okay. B. Number 2. A regulatory system that allows bacteria to choose glucose over lactose when both sugars. This is called the metabolite. No. I was going to say it's true, but it's actually called catabolite repression, not metabolite repression. All right. So the answer is false. Number 3. Attenuation control of tryptophan operon. The ribosome is stalled at the termination sequence during transcription and causing not termination sequence. No, ribosome prevents formation of the termination hairpin by binding to the polymerase. That's also not true. The CAMP molecule produces adenylate cyclase will bind to the CRP protein, and that's not about tryptophan operon. The polymerase is halted at the 3,4 termination hairpin if tryptophan level is high. That's correct. So the answer is D. Catabolite repression, attenuation. So number 4. There are 8 possibilities of transversions and there are 4 possibilities of transitions. You may want to be able to draw this. You draw A, T, G, C, Cs, and you can calculate. It's actually the other way. Oh well, yeah, this is correct. There are only 4 possibilities of transition, and there are 8 possibilities of transversions. Yeah, I will stop at that. So that's true. In a point mutation, how many nucleotides are usually involved? Well, I mentioned this in class, I think. When I studied, point mutation is single base, one nucleotide. But now, it seems that when you even have up to 5 bases, they're still considered as 5 point mutations, right? So 1, 1 and 2, 3, 2, 5. So the answer will be C, all of these. Next. Base pair additions and deletions that alter reading frame of all subsequent base pair triplets are collectively referred as frame shift because it changes the frame. So they are called frame shift mutations. C. Base pair substitution involving the replacement of purine with a pyrimidine. That is called a transversion. Changes between the same class, purine to purine or pyrimidine to pyrimidine, is called transitions. Base pair substitution in which purine on one strand is replaced with a different purine and a pyrimidine in a complementary is replaced with a different pyrimidine. That's called transition. Hope you didn't hear that. I just burped. In prokaryotes, genes with related functions often are present in coordinately regulated genetic units. Oh, this is called the operons. C. Sigma factor is a subunit of RNA polymerase, not B, an enzyme needed for transcription to proceed. It's not an enzyme. The place where RNA polymerase attaches. No, it's the place on RNA where RNA polymerase attaches. That's not true. That's not correct. So the answer is A, a subunit of RNA polymerase. So the answer is A, yeah? Question number 11. The tryptophan in the tryptophan. The method of gene regulation depends on whether the leading mRNA is allowed to fold. Correct. There is enough tryptophan in the cell. Correct. The ribosome is moving freely. Correct. RNA polymerase identifies the tripopron promoter region. No, that's not. So the answer is 1, 2, and 3, B. Next question. Tripopron. Next question. Which of the following best describes the lack operon positively control inducible? No, it's actually negatively control inducible. Yeah, so it's C. All right. Negatively control inducible operon. Okay, next question. Okay, this one. What type of regulation occurs when the product of the regulated gene is required to turn on expression? If it's required, that means it's a positive control. Because if you need to remove something like a repressor, so that's negative control. So this one is positive control. All right, so that's E. Next question. Where is it? Okay. The arabinose operon is an example of regulation by gene expression by an attenuation. No, false. No, attenuation is on for the tripopran where the mRNA is involved. The RNA is involved in ensuring that whether the termination happen is on or off. So that's so arabinose. Yes, there is modification of the structure of DNA, but not the mRNA. Okay. Arabinose operon. Gene regulation only occurs at the level of transcription. This is looking at my very first slide where it says gene regulation can occur at many different stages. Right? It can occur at translation, at the mRNA stability, the post-transcriptional regulation. So there's there are various steps that you can actually put gene regulation. So this answer is also false. The RC protein acts as both repressor and activator in the arabinose operon. This is true. This is one special property of the arabinose operon. Next question. Allolactose is a product of the reaction catalyzed by beta-galactose. Correct. Other than it's being broken up into glucose and galactose, if I'm not mistaken. Then one of the product is also allolactose, which serves as the inducer. All right. Next question. Lactose operon is expressed when lactose levels are high. No, it's wrong. It's correct. It's wrong because it is expressed. They need to meet two different conditions. Even if it's high in the cell, but if there's glucose, it's still not going to be switched on, right? So this is a false statement. It has to say it's high and glucose is absent, right? So this is false. Bacterial and archaeal cells are polycistronic, meaning that a single mRNA molecule often bears coding for transcript of few genes in tandem. This is true. This is correct. And finally, sigma factor have catalytic activity and help RNA polysaccharides in the promoter. Yes, true. Okay, so that marks the end of the quiz review. All right, so I hope if you have any questions, let me know. All right, otherwise, good luck for your exams. Bye bye.